All right, welcome to our notes on DNA. We're going to look at structure, function, and the history of DNA. All right, the first scientist we're going to talk about is Frederick Meissner. He was not born in 1869, but that's when his discovery took place. I'm going to give you a little bit of background on him. He was born in 1844 in Basel, Switzerland, to a wealthy and intellectual family. Both his dad and his uncle were also doctors. He wanted to become a priest because he was very, very shy. He didn't work well with people. He had some hearing difficulties. He had lost hearing from a childhood disease. But Dad said, no, you're going to become a doctor just like the rest of us. And that was how things went in that time. So he went through medical school. He was a really, really good learner, and he ended up studying tissue and natural chemistry. He didn't want to work with people like his dad and his uncle because of that shyness. So he went and started to do research. So what he actually started doing research on was leukocytes. So leukocytes are white blood cells. Leuko means white, and cyte means cell. And so the reason he chose that is because they're large and they were also easy to get because he worked near a hospital. So he would take their dirty bandages and the pus that was on that bandage. Those are extra white blood cells resulting from an infection. So he could take that pus and then kind of mix it with a couple other things depending on what he was doing, and he could look at just the white blood cells. He ended up discovering a substance that contained phosphorus and nitrogen, and he found that inside of the cell nucleus, and he was able to extract that, to take it out of the nucleus. This was the first time that was done. He called it nucleon because it came from the nucleus. Later it was renamed nucleic acid, and we know now that it's DNA and then also RNA, but they did not know, and he did not know, what it was at the time. He was just able to extract it. He did all of his research from using Darwin's theories of evolution and also a man named Gregor Mendel, who is the father of genetics, and we'll be talking about him later on. But he really didn't know what DNA did. It took another 49 years before people realized that kind of slimy substance that he took from the pus was holding all of our genetic material. I want you to keep in mind that he was only 25 when he made this discovery. Only 25. His work was not published till 1971. He did continue work on DNA or nucleon as time went through. He just loved his research. He got married in 1878, and then he became the professor of physiology at the University of Basel. So he studied, again, still DNA. He studied salmon. Being a professor gave him the opportunity to have more money to do his research, which is why he took the position. He was not a very good teacher. He didn't work well with kids because he was shy. He could not relate to them, and he was also a workaholic. But he did continue his position there for a while. He became known as the father of DNA. Ultimately, he did end up dying in 1895 from tuberculosis at the age of 51. So his discovery was he discovered nucleon, which is DNA and white blood cells. And it's important because he laid the groundwork for most of, actually all of our DNA studies that we have had in the future. So this is what he looks like, and this is the laboratory that he worked in. The second scientist we're going to look at is Rosalind Franklin. She worked with a man named Maurice Wilkins, and her discovery came about in 1952, but I am going to give you a little bit of background on her. She was born in 1920 in London to a rather affluent and influential Jewish family. She was exceptionally intelligent, knowing from a young age, around 15, that she wanted to be a scientist. She liked chemistry. In 1941, she graduated from Cambridge University with her bachelor's degree, and then she went to work for the British Coal Research Association, and she ended up writing a paper there which earned her her doctorate in 1945. From there, she started working in Paris studying X-ray diffraction. She worked on that for a while, really enjoyed it, and then moved to the King's College in London in 1951. She was offered and she accepted a research scholarship where she would be using her X-ray diffraction, studying DNA specifically. That's all she was going to study. And she worked with a man named Maurice Wilkins, and they did not get along. There were some personal conflicts. So he was a physicist, and he had worked in World War II. All of his research had gone to helping develop the atomic bomb, and he was not happy with that. He didn't like that his knowledge and research had gone to developing a weapon of mass destruction like that. So he was a little bit disillusioned, and he wanted to change sciences completely. So that's how he ended up in DNA and genetics and this X-ray diffraction. So like I said, they worked together, but they did not like each other. And in 1952, Rosalind Franklin took this picture. It is called Photo 51, and it shows this distinctive pattern of DNA. And that's what indicated the helical or that coiled shape of DNA that no one previously knew. In 1953, Maurice Wilkins went behind her back and disclosed her picture without her knowledge or her consent to Francis Crick and James Watson, which we'll talk about next. And that's what kind of solved their problems. From all accounts, she was gracious and she did not throw a fit. I mean, obviously, she probably wasn't happy. There's some people that speculate she didn't even know that her picture was the missing piece to Watson and Crick's research. She did leave King's College in 1953 because of some of the stuff that was going on and not getting along with Wilkins, and she went to Birkbeck College. So part of her leaving King's College and leaving her research scholarship, it meant that she could no longer study DNA. So she started studying the tobacco mosaic virus and RNA. RNA is another type of nucleic acid. It's similar to DNA. She started studying that, and in 5 years, she wrote 17 papers on viruses, which laid the foundation for all of our studies on virology, all the studies of viruses. Everything we know now, a lot of it began with her research. In 1956, she developed ovarian cancer. So it had to do when she was developing just this picture alone, just this picture. It took over 100 hours of X-ray exposure. So she was exposed to a lot of radiation over her career. She ended up dying at April 16th, 1958. She was only 37 years old. She did work up all the way through the end. She had 3 operations. She had chemotherapy, but she did love her work, and she worked all the way up through the end. Towards the end of her life, she did become friends with Francis Crick and his wife, who we are going to talk about in a second. I want to talk about this is all you need to write down, is that she showed the 2 DNA strands using X-ray diffraction, that there was actually 2 strands, and her work was used to describe the shape of that coiled dimension. So we're going to look at, We're going to look at James Watson and Francis Crick next, and their discovery came in 1953, but again, I'm going to give you a little bit of background. James Watson was born in 1928. He was 23 when he made this discovery. He was born in Chicago. He had 2 degrees in zoology, the study of animals. So one was his bachelor's and one was his PhD. He became interested in the study of genetics. So he started studying bacteriophages, which are viruses that invade bacterial cells to make copies of themselves or to replicate. He went to Denmark to continue his studies on genetics and to also get a little bit more information on chemistry because he wasn't very good at chemistry. In 1951, he saw Maurice Wilkins, the man that worked with Rosalind Franklin. He saw him in, I believe it was Naples, but it was in Europe, and heard him do a talk on the molecular structure of DNA. And after that, he was hooked. He wanted to solve that puzzle. Meanwhile, Francis Crick, who was born in 1916, was working in a lab in London and studying some genetics. But these two met up in the Cavendish lab in London, and they immediately hit it off, and they decided that they wanted to work together and they were going to build this model. A man named Linnaeus Pauling had just come up with a structure, but it was wrong. So they were trying to beat him to it. They used all the evidence that they could find, that they could collect about DNA over the many years, and they were building several models out of cardboard. Remember, there were no computers, so it was all 3D models. In 1953, it was Wilkins giving them the picture of Rosalind Franklin's X-ray diffraction, photo 51, that was that last piece in the puzzle, and it was enough information for them to figure out that the DNA was this double helix shape, that there were two strands, and that it looked like a twisted ladder. So they're credited with the discovery of the structure of DNA, and it showed how DNA was able to replicate itself by having that specific structure. After they made this discovery, Watson did go back to the U.S. He worked in a lab in New York. In 1968, he became the director. And then in 1988 to 1992, he was in charge. He headed the National Center for the Human Genome Research at the National Institutes of Health. And then he ended up returning to the same lab in New York and ended up retiring in 2007. He is still alive, but he is in his 90s. Francis Crick stayed in London and worked at Cambridge for about 20 years, and he made several major contributions to unlocking the genetic code. Then he ended up joining the Salk Institute for Biological Studies right here in La Jolla, California. It's a little bit south of us. There were lots of scientists that helped with these discoveries of DNA, but these are the three of the main ones. All right, this video right here, genes, DNA, and chromosomes, it's about 2 minutes. Because this is my video, you can't watch it here, and I'm not going to play it. So the link is in Haiku. Please do watch it. It has a lot of really easy information to follow. All right, let's review a little bit about cells and DNA. So what you should know, or hopefully are remembering, is that in your cell you have a nucleus, and the nucleus controls all of the cell's activities. It's the brain of your cell. Inside your nucleus is your DNA. That is the instruction manual for the nucleus and for your entire body. It holds everything. So we're going to look a little bit more in depth in that. Your DNA is really long. It's very thin, and it's coiled very tightly so that it fits in your nucleus. But to give you a little bit of an idea of how long your genome or your DNA is, if you typed 60 words per minute for 8 hours a day, it would still take you about 50 years to type out the human genome. It is that long, and you have it in all of your cells. Okay. All right, let's look a little bit at our DNA basics. First, DNA stands for deoxyribonucleic acid. Deoxyribo for the D, nucleic for the N, acid is the A. Deoxyribose comes from the type of sugar that's in there, and then DNA is a nucleic acid. It is located in the nucleus of all cells, most cells, pretty much all of them, and its function or its job is it holds instructions for all of the structures, all the chemicals, all the functions of the whole organism. It is your instruction manual. Everything your body makes, does, has, comes from these DNA instructions. So they're very, 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 very important. You don't have to write this down, but it's a little bit of information. If you compared human to human DNA, like compared my DNA to your DNA, or your DNA to your friend's DNA, it's 99.8% the same. So it's only that 0.2% that makes you different from other people. If you compare humans to chimpanzees, this is a chimpanzee right here, our DNA is 99% the same. And if you compare a human to a gorilla, it is 98% the same. And these are our closest living relatives. All right, let's look at structure. So DNA is made up of something called a nucleotide. So DNA, I talked about that being something called a nucleic acid, which is called a polymer. Poly means many, mer means parts. So this is made up of many parts, and those many parts are called nucleotides. So nucleotides are monomers. Mono means one, mer means part. Okay. A nucleotide is made up of three things, and I will show you a picture of this so it makes a little bit more sense. It's made up of a 5-carbon sugar, deoxyribose, which is again where we get the name for DNA. It's made up of a phosphate group and a nitrogenous base. If you hear nitrogen base or nitrogenous base, same exact thing. So here's a picture of that. Let's look at this side first. So if we have one phosphate, one sugar, it's 5 carbons, so that's 1, 2, 3, 4, 5. You would find a carbon on each point, and one nitrogen base. So on DNA, if we're looking at that, remember it's this double helix or twisted ladder. We've got the sides and the middle, and here is a nucleotide. Phosphate, sugar, nitrogen base. Phosphate, sugar, nitrogen base. So again, you could separate all of these, and it makes up, these are your nucleotides that make up your nucleic acid or your DNA. Let's look a little bit more at the structure. The backbone or the sides of DNA, so the sides of the ladder, are alternating sugar, again deoxyribose, and phosphate molecules. So I put a little ladder down here. Sugar is going to be our turquoise circles, phosphates are our stars. So if it's sugar, phosphate, sugar, phosphate, sugar, then the other side is going to be the opposite. So if this side starts with sugar, this side's going to start with a phosphate. Phosphate, sugar, phosphate, sugar. Okay, so they're going to be alternating. The steps or the middle of that ladder contain the code for all of the important information, and that's called your nitrogen bases. So there's 4 nitrogen bases. They are adenine, guanine, thymine, and cytosine. Adenine and guanine are something called purines. Thymine and cytosine are pyridamines. Okay. So if you look at this picture here, this is showing just half of a DNA molecule, which is one strand. So we have our phosphate and our sugar and our base, nitrogen base, so that's one nucleotide. And our bases are adenine, guanine, thymine, cytosine. And you can just call them A, G, T, and C. So in a full DNA molecule, you would have T right here, C right here, A right here, and G right here. And then this side would kind of be flip-flopped. And so it would be sugar, phosphate, sugar, phosphate, sugar, phosphate. So A always matches with T. Always, always, always. Think of them as puzzle pieces. So you have your purines and your pyridamines, and so one always fits with the other. T is always with A, G is always with C. This is base pairing, and this is what holds the 2 strands together. It's what holds those 2 DNA strands together, and they're held together by hydrogen bonds. T and G will never, ever, ever fit together. They don't like each other. They won't go together. Same with A and C. They just don't fit. So there's no what ifs. It just will not happen because they do not fit. It's TNA and C and G together forever. So a little bit of a recap. This whole DNA molecule, deoxyribonucleic acid, is called a nucleic acid. It is made of nucleotides. A nucleotide is made up of one phosphate, one sugar, and one nitrogen base. So when you put those together, this is one strand. This is your second strand. Base pairing, G, C, T, and A, that's held together by hydrogen bonds, and that's what holds these two strands together. And then your backbone or your sides are made up of sugars, phosphate, sugar, phosphate, sugar, phosphate, all the way down, sugar, phosphate, sugar, phosphate. So that's what makes up our DNA. And this is another short video, about 5 minutes, of what DNA is and how it works. I highly recommend you watch it. The link is in Haiku for you to watch. If you have any questions, please ask.